Lecture number 3 in our continuing series on drugs and human behavior. Today we're going to talk about pharmacodynamics, which is particularly important for drugs and human behavior and psychopharmacology because we have to really understand how drugs interact with receptor subtypes within the brain, and so we're going to spend a little bit of time talking about that. So what we're going to talk about in this lecture is a quick introduction to what we mean by pharmacodynamics. Talk about receptors for drug action, the different receptor structures, talk about dose-response relationships, drug safety and efficacy, talk about what's called the LD50. This is the dose at which 50%, which is lethal for 50% of the population. Talk about the therapeutic index, drug interactions and side effects, and then finally finish up with a discussion of placebo effects and what are called nocebo effects, which are negative placebo effects. But to start off, when we're talking about pharmacodynamics, this is in contrast to pharmacokinetics. Pharmacokinetics is what the body does with drugs. Pharmacodynamics is the study of what a drug does to the body. So how does the drug interact with different cells, different receptors, etc. So here we're primarily going to be looking at how drugs affect different receptor subtypes on neurons. That's the entire field of psychopharmacology hinges around this particular question. So when we start talking about receptors for drug action, we're talking about large molecule sites where naturally occurring compounds, neurotransmitters or modulators, what we would call an endogenous neurotransmitter, produces some biological effect. So these large molecule sites are where dopamine would bind, where serotonin would bind, that sort of thing. There are hundreds of different receptor types known. We talk about neurotransmitters that can be specific to certain receptors. So we talk about dopamine. There are 4 or 5, several different kinds of dopamine receptors and dopamine molecules. And the same thing is true of serotonin. There are several different kinds of serotonin. So neurotransmitters can be specific to certain receptors, but a drug may be more specific than the actual endogenous neurotransmitter. So while an endogenous neurotransmitter, that is a naturally occurring neurotransmitter, may have wide effects, drugs might actually have much more localized or specific effects than those endogenous neurotransmitters. These drugs we're talking about form what's called the reversible ionic bond with specific receptors. That is, it binds for a minute, forms that ionic bond, has its effect, and then releases and is metabolized out, and just like any other neurotransmitter. Binding to a receptor results in one of 3 different kinds of actions. Binding to the site of a normal endogenous neurotransmitter initiates a similar cellular response. This is what we call an agonist action. This is where a neurotransmitter is having the exact same effect a drug is having the exact same effect as a neurotransmitter. So it binds to that same site, causes the same kind of cellular response that we would expect from the endogenous neurotransmitter. Binding to a nearby site that facilitates that transmitter binding is called an allosteric action. And so a drug that has an allosteric action is one that will facilitate a neurotransmitter. And then finally, binding to a receptor site, thereby blocking access to the transmitter binding site, therefore blocking a neurotransmitter from having an effect or some other drug from having an effect, is called an antagonist action. So an antagonist is going to block the effect of some neurotransmitter. So agonist actions increase the effectiveness or the use of a neurotransmitter. Allosteric actions facilitate neurotransmitters, whereas antagonists block the action of neurotransmitters. There are, of course, a variety of different kinds of receptor structures. We talked a little bit about this in previous lectures. Ion channel receptors are those in which activation opens a channel, allowing the flow of ions to trigger or inhibit neural firing. So we looked at these previously. An ionotropic receptor is one in which neurotransmitter binds to it, opens up that ion channel, and causes that neurotransmitter or that neuron to fire. Benzodiazepines are what we call a GABA receptor allosteric agonist. A mouthful, I know. But essentially, here's what's happening. They bind to nearby sites and facilitate the functioning of GABA, which is gamma-aminobutyric acid. That then floods the neurons with chloride ions and inhibits neural actions. We didn't get into the details of how action potentials work, But chloride ions are one of the things that actually reduces the likelihood of an action potential from being fired because it changes the resting potential. It essentially hyperpolarizes a neuron and so inhibits that neuron from firing. So benzodiazepines bind to nearby sites and facilitate GABA's functioning, which is why they're used as sedatives, anti-anxiety drugs, cause amnesia, and are also used as anti-epileptic drugs. So if we take a look at that receptor, you can see there is a receptor subtype that has a place for benzodiazepines to have their influence. For GABA, this is a similar receptor that is influenced by ethanol as well as neurosteroids and propofol. Important to understand that we want to be very careful of any mixing of benzodiazepines and alcohol because they affect the same kind of receptors and cause very strong inhibition of neurons and can of course lead to things like difficulty breathing. So that's the benzodiazepines effect on this kind of ion channel receptor. To give you an example of an antagonist action, flumazenil actually binds to that benzodiazepine site but doesn't interfere with GABA. So it essentially blocks the benzodiazepine from having any effect. So flumazenil is actually a drug that will instantly reverse an overdose of a benzodiazepine. So this is a drug that anesthesiologists would always have on hand in the event that a patient overreacted to something like midazolam. So flumazenil actually completely reverses the effect of benzodiazepines. So it's technically a benzodiazepine antagonist, which is used to treat benzodiazepine overdose. So it's pretty remarkable that it blocks that benzodiazepine site, thereby no longer facilitating the influence of GABA, and so completely reverses the effects of a benzodiazepine. G-protein coupled receptors are these that we talked about that don't open up ion channels but cause other secondary actions. So they induce the release of intracellular proteins and trigger secondary messengers. These are also called metabotropic receptors. They control many cellular processes that is they can control ion channel function, energy metabolism, cell division, differentiation, and neuron excitability. So they control a lot of different processes within a neuron. So what happens with a G-protein receptor is the neurotransmitter binds to it. Then that causes the transmitter binding to activate the G-protein receptor. That then causes some activation of a subunit of an ion channel, which then causes some delay. So this is a slower process and a much different process from an ionotropic receptor. So to give you an idea how this process works, we might have a process starts when, say, a hormone or neurotransmitter attaches to the receptor. The receptor shape changes, and the three-chain G protein on the inside of the cell membrane, that component of the G protein is released and directly or indirectly activates an ion channel. Those ion channels open. Alteration of enzyme activities or changes in gene activation may be produced, and then we get communication between neurotransmitter receptor complex and the intracellular enzymes that produce the ultimate biological response. So it's a lot of different pieces to this puzzle that happen when G-protein coupled receptors are involved. Carrier proteins are those that bind to neurotransmitters to transport them back into a presynaptic neuron. Remember we talked previously about reuptake, which is the process of taking neurotransmitters back into the presynaptic neuron. And carrier proteins are responsible for that. So many drugs will actually block the carrier proteins for some specific kinds of neurotransmitters. So for example, we're going to talk about what are called selective serotonin reuptake inhibitors that block the reuptake of serotonin into the presynaptic neuron. Enzymes are of course present in the synapse, and their function is to break down neurotransmitters in the synaptic cleft. Increasing these enzymes causes an inhibition by drugs. The inhibition by drugs can increase transmitter availability, so inhibiting enzyme functioning increases transmitter availability. So for example, a monoamine oxidase inhibitor will block the effect of monoamine oxidase, thereby increasing the availability of dopamine and serotonin in the synapse. We talked already about irreversible acetylcholinesterase inhibitors. These are used as pesticides in nerve gases. Essentially what they do is they keep acetylcholinesterase from breaking down acetylcholine in the synapse and thereby causing muscle fibers to constantly fire, resulting in paralysis and death. Monoamine oxidase inhibitors, as I was just mentioning, inhibit the breakdown of norepinephrine and dopamine and are often used as antidepressants. There are acute and chronic receptor effects that are important to understanding the long-term and short-term effects of drugs on neurotransmission. So when a psychoactive drug binds to a receptor, it produces an immediate response, so that receptor immediately responds. When a drug is given over a longer period of time, it produces long-term changes in the properties of receptors. You can get what we call downregulation, which is desensitization of neurons, or upregulation, which is supersensitivity to neurons to certain neurotransmitters. An example of this is long-term use of nicotine. Nicotine actually causes an alteration in the sensitivity of GABA. It actually downregulates GABA. But when you take away nicotine, you then end up with a hypersensitivity. Without nicotine, GABA is upregulated. So during nicotine withdrawal, you have upregulation of GABA, and as a result, a great deal of neural inhibition. And in fact, some of my research has shown that this upregulation of GABA during nicotine withdrawal is responsible for significant memory reductions during that withdrawal period. So to take a look at how this works, you can get long-term downregulation, which is an agonist effect, or long-term upregulation, which is an antagonist effect. So those are important to understand the long-term influences of drugs, and also what can happen when someone tries to withdraw from a drug like nicotine. To determine the effect of a drug, we often have to study several doses and measure the change in some response. And we call this a dose-response relationship. So it's the relationship between how much of a drug is given and what kind of response we get. And we usually call this a dose-response curve. In general, dose-response relationships are not linear. So when we start to look at these dose-response curves, we can actually look at two different kinds of dose-response curves. One which compares the intensity of a response to its dose, and we see that on the right, the percentage of the maximum effect of a drug dose. So for example, if we were looking at a sedative, the maximum effect of a sedative is going to be unconsciousness. So but maybe we don't want unconsciousness, we just want alleviating anxiety. And so we can look at this dose-response relationship between the dose and the intensity of a response. Similarly, we can look at the dose and percentage of subjects that respond to that particular dose, because everyone's a little bit different. Not everyone's going to respond the same way to the dose. So we talk about the potency of a drug being associated with this dose-response curve. So the lower the dosage and the higher the effect, the more potent a drug is considered to be. And we'll take a look at some comparisons of drugs here in just a moment. So this dose-response curve gives information about three main variables. The potency of a drug, which is how well drug molecules attach to their sites of action, that is at the receptors. So a highly potent drug is going to attach and have a great deal of effect. You get maximal effect from less drug. So more potent drugs usually attach better than less potent drugs, so they bind better, and they bind more tightly than less potent drugs. So binding has to do with the affinity. The more potent a drug is, it has a greater affinity for its receptor. So you can just think about this as they fit together very well and they stick together, so there's a great deal of binding and affinity between neurotransmitter or drug, and its receptor site. A less potent drug has less affinity for its receptor, does not bind so tightly, so it can be more easily knocked off the receptor, and so isn't nearly as potent. So potency is an important component of understanding dose-response curves. So different drugs may bind to the same receptor, but with different affinities. So when we talk about the potency of amphetamines or the potency of different benzodiazepines, They bind to those benzodiazepine receptors at different potencies. The second characteristic illustrated by the dose-response curve is its slope. This refers to the mostly linear central part of the curve, how sharply the effect changes with each change in dose. If a small change in dose produces a large change in effect, the slope is steep. If a large change in dose produces small changes in effect, the slope is shallow. And that's really important information because it tells us a lot about how increasing the dose of a drug may or may not have any effect on behavior. So when we're talking about something like caffeine, it has a relatively shallow or sort of relatively small slope. When we're talking about amphetamine, it has a very steep slope. Efficacy, then, is the final information we get from a dose-response relationship. This is the maximum effect obtainable with additional doses producing no effect. Some drugs may be potent, but they might never be able to produce a peak response no matter how much is given. A drug that is more efficacious can produce a greater peak or maximum effect than a drug that is less efficacious. So if we're talking about trying to limit pain, for example, and we'll talk about that here in a few lectures, a more effective drug can reach a greater amount of pain relief than a different drug. So aspirin is only going to get you so far, whereas morphine is going to cause probably the greatest amount of pain relief that you can get. So to show you different kinds of drugs that have different dosages and different potencies and different efficacy, if we look at methamphetamine, D-amphetamine, and caffeine, a few milligrams of methamphetamine cause pretty sharp responses, and it doesn't take a lot more methamphetamine to get to that maximal response. You can get to that same maximal response with D-amphetamine. It just requires a higher dose. So D-amphetamine is just as effective, but requires a higher dosage, so it's not as potent. Methamphetamine is much more potent. Caffeine is not nearly as effective if we're talking about the kind of response, alertness response. So caffeine, you're only going to get so far with caffeine doses, and you can see there's a pretty slight slope. Increasing, it takes a lot more caffeine to get even a slighter response, whereas just a little bit of amphetamine gets you a lot further. So this demonstrates both potency and efficacy. Similarly, if we look at heroin versus morphine versus aspirin, heroin and morphine have equal efficacies for pain relief, whereas aspirin is a limited amount of pain relief. Heroin and morphine have similar efficacies. Heroin is much more potent than morphine, and obviously aspirin not nearly as effective and has a much shallower slope. So increasing amounts of aspirin aren't going to get you much pain relief. So to summarize the receptors for drug action and some issues that we talked about previously, remember that binding to a site of a normal endogenous neurotransmitter initiates a similar cellular response. We call that an agonistic action, whereas binding to a nearby site to facilitate neurotransmitting is called an allosteric action, and binding to a receptor site blocking access is an antagonistic action. So that is a quick review of the issues associated with different types of receptors. If we look at different efficacies and different dose-response relationships at antagonists, agonists, and partial agonists, full agonists, and inverse agonists, you can see you get changes in responses based on partial agonists and partial agonists and complete agonists. An antagonist is going to block response from that particular neurotransmitter, and an inverse agonist is actually going to reverse or reduce the functioning in that situation. So an antagonist drug by itself produces no response. It's possible to determine if a drug has an antagonistic action by seeing if it reduces the effect of the agonist. So this is, again, a dose-response relationship, as an antagonist drug produces no response, but it can actually eliminate the response of a different drug, thereby having that kind of secondary response. There are irreversible or non-competitive antagonists that can cause a downward shift of the maximum with no shift of the curve on the dose axis. So this just simply limits the efficacy of whatever drug it is, or whatever neurotransmitter is an antagonist of. So a brief summary. If you look at an agonist dose with the agonist alone, if we have an agonist plus a competitive antagonist, it requires a larger dose for the agonist drug to have an effect. If we have the agonist alone, and then again an agonist plus an irreversible antagonist, it's going to reduce the maximal effectiveness. So a competitive antagonist is going to just simply require a larger dose to get to the same effectiveness. An irreversible antagonist is going to reduce the maximum effect possible. So to give a summary of these issues, agonists bind to receptors to produce a functional response. Agonists can be full, partial, or inverse. Antagonists block or reverse the effect of agonists. And antagonists can be competitive or non-competitive and irreversible. The final issues I want to talk about have mostly to do with drug safety and drug interactions. For any drug to have FDA approval, it must be proven to be both effective for its purpose, its intended purposes, and it also, of course, has to be safe. So understanding how a drug is effective, something we've already talked a little bit about. We're going to talk about the standards for that and also standards for safety. So when we talk about effectiveness, we actually talk about what's called the ED50, which is the effective dose for 50%. So the ED50 is the drug dose that produces the desired effect in 50% of test subjects. The LD50 is the lethal dose for 50% of test subjects. We're going to talk more about that here in just a minute. The lethal dose is tested using experimental animal models to try to determine that lethal dose. We obviously don't test lethal doses in humans. We'll talk more about the therapeutic index in a minute, which is the ratio of the lethal dose to the effective dose. And we'll talk a little bit more about what that means here in just a minute. But to give you an idea of which drug is safer, for example, we can look at the dose-response curve and where the lethal dose is. So instead of maximum effectiveness, we look at the percentage of animal deaths at the maximum, and so 100% deaths are at the top of the curve, and so the point at which a drug is causing 50% deaths is the LD50. So you can see that drug B is in many ways safer because it can be given in higher doses without causing as many fatalities. So drugs have many effects, of course. There's usually a prominent desirable response as well as other actions that are undesirable. Some side effects, of course, can be lethal, things like inhibition of breathing or respiration, cardiac arrest, toxicity, that sort of thing. So the dose of a drug that will be lethal in 50% of subjects is called the LD50. And it's really important to understand the relationship between the therapeutic effects of a drug and its possible lethal effects. So for example, we're going to talk about drug overdoses, particularly when we talk about opioids and overdoses for opioid analgesics. We also talk about overdoses with barbiturates because they do have very lethal effects in inhibiting breathing and causing significant death and harm that way. So we're going to talk a lot about this in some areas. Most of the drugs we're going to talk about, the lethal doses are so high that there's very little chance of a fatal overdose. The primary drugs that cause fatal overdoses are those that have effects on breathing or those that can be cardiotoxic. So cocaine, methamphetamine, generally people don't technically overdose. They just simply have a heart attack from too much use of a drug. So when we talk about the therapeutic index, this is the ratio between the lethal dose and the effective dose. So the greater the therapeutic index, the safer the drug is. So the difference between the desired effect and its undesired or lethal effect is larger. So the larger that therapeutic index is, the safer a drug is. If two drugs produce a therapeutic effect that is desirable, the better drug, all things being equal, would be the one with the larger therapeutic index. To be safer, the TI, the therapeutic index, is actually calculated using the ratio of 1% to 99%. So the lethal dose for 1% of the population versus the effective dose for 99% of the population. So it's a much safer estimate. So the lower the ED50, that is the effective dose, the greater the potency. So the lower the effective dose, the greater the potency, but the lower the therapeutic index, the lower the safety. So if we look at something like percentage of subjects showing response to a couple different drugs, the drug on the left in orange has an effective dose of 10 milligrams and a lethal dose at 50 milligrams, whereas the drug on the right in green has an effective dose at 50 milligrams and a lethal dose at 100 milligrams. So the therapeutic index is 5/50 versus 50/95. So, sorry, that's not right. 100/50 versus 50/10. So that's the therapeutic index. We'll talk a little bit more about that later on when we start talking about what kind of doses to talk about. The last thing I want to talk about have to do with drug interactions and safety, and then we'll finish up with placebo effects. Polypharmacy is where we combine medications to improve a therapeutic outcome. So oftentimes 2 drugs will be given in combination to improve a therapeutic outcome. So oftentimes we are currently advising some patients to combine a second drug with an SSRI antidepressant to try to increase the efficacy to get at their depressive symptoms. We try to avoid what's called pharmacodynamic redundancy, where drugs have the same or overlapping mechanism of action, unless one is improving the functioning of another. But when they have the same overlapping mechanism of action, you are really battling, the drugs are battling against one another for one to be effective. Remember, if they have the same neurotransmitter receptor, they're going to be competing with one another to bind that receptor. So there are several principles to reduce the sort of likelihood of irrational polypharmacy. There are reasons to take drug combinations, but you want to watch out and make sure that's done carefully. You want to be first careful about pharmacodynamic interactions. 2 drugs that have the same or overlapping mechanism of action. Pharmacokinetic interactions. 1 drug can affect the other's absorption, distribution, metabolism, or excretion. You want to watch out for inadequate dosing. Doses are often too low because of side effects. And then watch out for clinical evaluation, or you want to make sure that you get clinical evaluation and oversight. Should always be ongoing. So you want to watch out and really be careful about how these things interact with one another. And there are some good tools for doing that. There are actually apps you can download to your phone to enter in your prescriptions, so that then if you're out and you need a cold medicine or something and you want to see if it interacts poorly with drugs that you take, you can just enter that into that app and see if it's going to interact. So I highly recommend those. We want to obviously avoid drug toxicity. All drugs can produce harmful effects as well as beneficial ones. Most drugs exert effects on several bodily functions. In general, we're often worried about respiration, particularly with psychoactive drugs, and also liver functioning, because of course all drugs have to be metabolized out through the liver, and so we really want to watch out to make sure that they're not having toxic effects on the livers. Of course, important thing to watch out for too are drug allergies. This can take on many forms. This will differ from normal side effects. Allergic drug reactions can include, of course, severe drugs like anaphylactic shock, or severe reactions like anaphylactic shock, where a person stops being able to breathe, rash, other side effects, dizziness. All of these kind of things can be caused by an allergy to a drug. And so you want to really watch out for that and be very careful and mindful, and just pay attention when you take a new drug to look out for these kind of side effects. There are a couple of resources you can look at. You can go to drugs.com and look at drug interactions, or the Hippocrates app is a terrific one. It's designed for physicians, but it works very well for doing things like drug interactions. So I highly recommend those. Final note in talking about pharmacodynamics, most studies, any good drug study is going to use a double-blind, randomized controlled clinical trial. It's double-blind because one group has placebo, one group will have the drug, and we have to look carefully at how these trials can misrepresent placebo effects. And so noticeably, placebo effects in antidepressant studies seems to have risen dramatically over the last 20 years. And so double-blind tests with the placebo run-in period might eliminate much of the placebo effect. Essentially what that is everyone's on a placebo for a short period of time, and then one group gets switched in to the drug trial if they do not respond positively to the placebo. So every patient is administered a placebo for a week. Those who respond significantly during that first week are then removed from the study because they are having such a strong placebo effect. There are lots of individual differences in these issues. Interestingly, lots of drugs some drugs show stronger placebo effects. Alcohol has a high placebo effect if people believe they've had alcohol, as do painkillers. So the take-home message here is placebo effects show individual differences, and you have to really watch out for that. And so eliminating those individual differences is an important part of doing an effective pharmacology study. Last point I want to make on placebo effects is you can actually get what are called nocebo effects. This occurs as a result of unintegrative negative suggestions by health professionals when informing patients of possible complications of proposed treatment. Essentially, what happens is the physician says, this might happen if you take this drug, and it's a self-fulfilling prophecy. The individual ends up with that side effect as a result of a placebo effect. In this case, it's not a beneficial effect, it's a negative effect. And so that side effect occurs not from the drug itself, but from the suggestion of that. And so it's something to really watch out for. It's obviously difficult to weigh the best way to approach these issues, but just simply be aware that that might happen. So that is an introduction to pharmacodynamics. Important to understand how drugs interact with receptor types, the ways in which they interact, and of course, how that influences dose-response relationships, etc. Thank you.